Before knowing the procedure, we'll know about the introduction first. Accurate measurement of glucose in body fluid is important in diagnosis and management of diabetes, hypoglycemia, adrenal dysfunction, various other conditions. So these are the importance or application of glucose test, where the accurate measurement of glucose in our body fluid, basically blood, is very, very important, which help us to diagnose and manage the disorder like diabetes and hypoglycemia, where the glucose level decreases, adrenal dysfunction, where we can say the adrenal gland, where there is a abnormalities, and various other conditions such as when we go for surgery. So before surgery, the doctor asks that do you have diabetes, so that can be uh, confirmed by glucose test, right? And here the clinical significance of the glucose test is the increased level of glucose may be seen in diabetes mellitus, the patient who have a diabetes mellitus, where the insulin is not produced properly, or maybe the insulin which is produced might be dysfunctional insulin, because the insulin may not be able to bind to the proper receptor. So the receptor may be abnormal, or maybe there will be the insulin insufficient production. Patient receiving glucose containing fluid intravenously. So those patients who receive glucose intravenously, okay. So in such patient also, for a short period of time, the glucose level might be increased. The patient who are under stress or severe stress, so their glucose levels are usually increased. Cerebrovascular accidents. So when there is a cerebrovascular accident, so in such condition, the glucose level in blood usually comes higher level. Hyperparathyroidism, which is the hyperactivity of parathyroid gland. The parathormone is produced in large amount, and we don't know actually how it increase the glucose level in the blood, but it has observed that patients suffering from hyperparathyroidism usually have a higher level of glucose in their blood. Pancreatitis is an inflammation of the pancreas, where the pancreas actually get inflamed, or the pancreatic cells are usually damaged, and in such condition, the pancreatitis condition, glucose levels are higher in the blood. And the last, the renal failure, when the kidney stops functioning. So in such condition, also the glucose levels comes higher level in the blood. I hope you are understanding. आप लोगों को समझ में आ रहा है? Yes. Are you able to understand? Just give me your feedback. The team has joined. Are you understanding? In what condition the glucose becomes higher level in blood? So we have yes. Lisa has given the message. Yes. Okay. So coming to the next slide, let me know if you can see the slide properly. Okay. Can you see the slide?
Yes, we can see.
Okay. So now let's know the another clinical significance of glucose test, where glucose level in the blood may be decreased. So decreased level of glucose can be due to insulin administration. So some patient might be taking insulin in the blood. Okay. So excess level of insulin dose may decrease glucose level in the blood. Similarly, another case is insulinoma, which is a tumor-like or cancer-like condition, where the insulin-producing cells of the pancreas become cancerous, and it produces abundant amount of insulin, and because of the action of insulin, glucose level in the blood decreases. Third, inborn error of carbohydrate metabolism. So this is a genetic defect in by birth defect, where the carbohydrate metabolism, there is some enzymatic deficiency, or maybe there is a condition where the glucose might be produced in the large amount, or maybe the glucose may not be utilized. So in such conditions, glucose levels can be, decrease in the blood. Okay, so there will be excess glucose utilization, or there may be no glucose synthesis, right? So this is an inborn error of metabolism. In such condition, glucose level decreases. Similarly, fasting condition, like when we do fasting because of some religious, like if Muslim do fasting, Hindu do fasting. Okay, so fasting is a trend in the religion, and the Christians sometimes also do the fasting. So this is the fasting is a religion trend, and when they do fasting, so there is a prolonged gap between the food intake. And what happens? This prolonged gap between the intake of the food usually decreases glucose level in the blood. Okay, and hypothyroid is where the thyroid gland function less than the normal what it function. Similarly, hypopituitary is where pituitary gland function less than the what it usually function. So in hypothyroidism and hypopituitary, in both conditions, it has been observed that there is a glucose level in the blood usually decreases. And lastly, extensive liver disease. When there is a severe liver disease, okay, so in such condition also, it is observed that the glucose levels in the blood decreases. So we studied the two conditions where glucose levels might be increased and where glucose levels might be decreased. Okay, so these are the clinical condition, and because of this significance, we do glucose test in the lab. Okay, let's know about normal range. Normal range of glucose is the fasting level of glucose is 70 to 110 mg per day, and if we talk about this 2 hours postprandial, this is 2 hours postprandial, right? 2 hours postprandial means after 2 hours of the intake of the food, the glucose levels in the blood becomes up to 140 mg per day. Okay, so it's up to 140. It can be above 110, but the upper limit is up to 140 mg per day. Urine glucose, usually there is no glucose in the urine normally, and CSF glucose is 60 to 90 mg per day. Okay, so to express the result in millimole per liter divided by 18. In some of the laboratories, they give the result of the glucose in millimole per liter. Okay, but some of the laboratory you can see they give the result in mg per day. Okay, so it depends upon the laboratory to laboratory, and what happens? If you want to, if you have the value of mg per dl, and if you want to convert mg per dl into millimole per liter, so this is the formula given to express the result in millimole per liter. We need to divide by 18. So when you divide by 18, we get the value in millimole per liter. And this is the molecular weight of glucose 180. So you need to remember this can be asked in the viva sometimes. Now let's know about the principle that the Trinder's method. What is the principle? Here, glucose in the sample is oxidized to yield gluconic acid and hydrogen peroxide in the presence of glucooxidase. So glucose oxidase is the enzyme. Here you can see this is the glucose oxidase, and what it does that the beta D glucose, this is the glucose present in the sample, and the glucose in the sample in the presence of water accretion, and this is the enzyme. What it does that it converts glucose into gluconic acid and mix water here. Hydrogen peroxide here, and after that the hydrogen peroxide react with the phenol and 4-amino antipyrine to catalyze by the reaction peroxidase. So it produces red dye or water. Okay, so here the color which is a complex which is produced is directly proportional to the concentration of glucose in the sample. Okay, so what we say that when the glucose concentration is higher, so the color which is produced, the intensity of the color is proportional to the concentration of glucose in the sample. This is the principle. Here we have two types of enzyme in the sample. So in the reagent, two enzymes are glucose oxidase and peroxidase. Glucose oxidase catalyze one of the specific reaction where glucose is converted to gluconic acid and hydrogen peroxide, and the peroxidase usually catalyzes the coupling of 4-amino antipyrine with phenol to produce the color complex, This is known as quinoimine color complex. Okay, so this quinoimine is a color compound. It's a red color compound, pink color compound, and the intensity of the color is proportional to the concentration of glucose in the sample. So we studied the principle. Hope you have no doubt. Now we require reagents, working reagents, which has the composition of it contain phosphate buffer, glucose oxidase, peroxidase, or amino antiphylate. So these are the ingredients present in the working reagents. Reagent one, and the reagent two is a glucose standard. The concentration is 100 mg per dl. Now, material required is photometer, that is a colorimeter, which is adjusted on wavelength of 540 nanometer. We require cuvette of 1 centimeter in length, and then water bath we require at 37 degrees Celsius for incubation. And we require automatic pipettes, what we call micro pipettes sometimes. We require test tubes and racks, and finally the disposable tips, which can be used for the different sample, different standard, different reagents. So these disposable tips are also required. So sample should be collected as a blood collected in a sodium chloride vial. So this is the gray cap, vial. So here you can see it is in sodium chloride and potassium oxalate. The mixture of sodium chloride and potassium oxalate. So is taken as a vial to collect blood, and sodium chloride is an inhibitor of glycolysis, and it preserves the glucose present in the blood. So because of that, we consider sodium chloride vial as a one of the preservative for blood glucose test. So blood should be collected in sodium chloride vial. Okay, you have to remember this. Why blood should be collected in sodium chloride vial? Because sodium chloride, the chloride basically inhibits glycolysis. It preserves the glucose in the sample. Now, general system parameters, like when we have a let's say analyzer. So here we need to calibrate the analyzer with this information. Reaction type should be endpoint, wavelength 540 nanometer, temperature 37 degrees Celsius, incubation we need to incubate for 10 minutes, and the incubation time is 10 minutes. And reagent volume we require 1 ml, sample volume we require, this is what, 10 microliter, and then standard concentration we have to fit 100 mg per dl. Zero setting should be done with a blank, and then light length is 1 centimeter. So this is the general system parameter that we need to remember about this glucose test. Now, procedure is, if you have to take a test tube, label it as B for blank, S for standard, T for test. Then we have to add 1 ml of working reagent in each test tube. Sample we have to add 10 microliter in T. Standard we can add 10 microliter in standard tube, and the water we can add 10 microliter in blank. We need to mix well, incubate for 10 minutes at 37 degrees Celsius, not at room temperature. We can just let me rectify this. This is at 37 degrees Celsius, right? 37 degrees Celsius. This is at 37 degrees Celsius at for 10 minutes, right? Are you getting my voice? Hello, students. Surprise.
I can hear you, yeah.
Yeah, Martin, yeah, okay. So I just wanted the feedback that you were able to hear me. Now we need to mix well. We can mix all the ingredients separately in each test tube. Then you can incubate for 10 minutes at 37 degrees Celsius. And after incubation, we can take the absorbance of standard, absorbance of test against the blank. So we can calibrate the colorimeter with the blank, and it should be measured. The absorbance will be taken at 540 nanometer within 15 minutes of the the incubation time. Now calculation we can do. Calculation is absorbance of test divided by absorbance of standard multiplied by 100. 100 is standard concentration. So now linearity is this procedure linear up to 600 mg per dl. When the sample contains glucose up to 600 mg per dl, it can give the results. But when the value exceeds 600 mg per dl, then we need to dilute. The sample above the concentration should be diluted the normal saline, and this should be multiplied by proper dilution factor. Okay, so we can multiply if the value is more than 600 mg per dl. So factor we can calculate what is the factor of dilution, and accordingly you can multiply. Okay, we have come to the end of slide. you have any question, please ask me. Yeah, now we are open for discussion here.